that such a circumstance existed, that your son or daughter could be diagnosed with an awful disease, which the medical world knows very little about, and that there could be no treatment or therapy or anything anyone could even try in order to help them. We founded the Yaya Foundation with a group of parents who have all had to deal with this reality, but who can also imagine a world in which a different reality exists for children and families affected by 4-H, where there are therapeutic alternatives available, thanks in large part to all of your great work. The Yaya Foundation is the only organization in the world devoted to fighting for people affected by 4-H leukodystrophy. And we do that by accelerating discovery of therapies that will enable people affected by 4-H to live longer, better lives, and by providing educational and emotional support to affected families. To accelerate therapeutic discovery, we've launched the 4-H Leukodystrophy Collaboration Network. The 4-H LCN is a global cross-disciplinary community of patients, researchers, and clinicians working together to discover therapies. And I'm excited to see so many of our wonderful collaborators here on this call today. Together, we leverage the perspectives and expertise of our community to identify the highest impact research questions to answer, find the right researchers to answer those questions, fundraise aggressively to support the work, and then procure samples and enroll patients in studies, execute studies, analyze data, identify treatments, and disseminate knowledge. We hosted our first working meeting of the 4-H LCN last November, recently released version 1.0 of our community's research roadmap, and have raised nearly $1 million to support the 4-H LCN and the work it prioritizes. We look forward to hosting global scientific seminars like this one on a regular basis going forward, and we very much look forward to continuing collaborating with all of you. We also look forward to sharing news about the 4-H LCN's projects and our next convening over the course of this year. Before I ask Valerie to introduce Professors Fribourg and Vannini, just a few thank yous. First, thank you, Professor Vannini, for agreeing to be our inaugural speaker. I couldn't think of a more interesting or appropriate topic to start our seminar series with, and can't wait for your talk. And thank you, Professor Fribourg. You have been a great partner since the founding and launch of the 4-H LCN, and we thank you for all of your wonderful collaboration and especially your work bringing this seminar about. Thank you to all of our 4-H LCN collaborators joining us today from across the globe, to all of our supporters and friends who are here, and to the Chan Zuckerberg Initiative and the Rare as One Project for supporting our work. A special thank you to our small but very mighty and in fact amazing Yaya Foundation team, Valerie Greger, our Science Director, and Christina Butterworth, our Managing Director. And most of all, thank you to the parents that are joining us for today's call. So with that, I'll hand it off to Valerie to introduce Professors Fribourg and Vannini. Valerie, thank you.
Thank you, Ron. Welcome, everybody, and I'm glad we are here. Ron has said it all. I mean, this seminar series is sort of to further our goal to have communication, collaboration in the scientific community, because that is one way that we heard that a foundation for rare disorders can support research and further research, and that's why we're here. And so it was very exciting when early this year, Sebastian, emailed us and said, "Hey, did you see there is this cool paper coming out about the structure of POL III, and there's actually another 2 to follow." And Sebastian, who is of course part of our initial research network and has participated in a long time, has mapped various or modeled mutations in 4-H genes long before the human structure was solved. So he knows everything about it. And so that's why he said, "Well, can I introduce you to a paper?" And I said, yes. And so this idea about the seminar came about. So I just want to say a little bit. So there were 3 publications, one from a laboratory in Dr. Professor Wang's laboratory in Shanghai. And then, of course, the group around Christoph Müller at the EMBL in Heidelberg. And last but not least, Alessandro's and a group together with a group in Regensburg who published these exciting results about the structure of the human polymerase III complex. So a little bit about Alessandro. Alessandro joined the Institute of Cancer Research in London as a team leader in 2012, which seems like a long time ago. And he became the head of the structural biology unit there in 2019. He has also, as he said, just joined, become a professor there, and is also now has returned to Italy at the Human Technopole Institute in Milan. But he's sort of, as I said, runs 2 labs remotely. So this is pretty challenging. Anyway, without further ado, Alessandro, I hand it over to you. Welcome, and I hope we have all fun and an interesting discussion. Thank you.
Thank you, Valerie, Sebastian, Ron, for the introduction and for having me here today. I must say, considering how many structural studies have been coming out for concerning human polymers in particular, must be that I've been probably the fastest one at replying to emails, etc. No, I'm just kidding, but I mean it's really great to see that there are so many groups interested in RNA Polymerase III, which was considered really to be a niche field. And as Ron already has described, this has actually a really important impact on well-being and impact really, especially very young patients. And so it's really important to be all here. It would be much nicer to be sitting, in a place and be in person, but this is the reality we live these days. So we try to get the best out of it. And I hope I will try in the next half an hour to summarize a little bit what we are interested in general, trying to touch upon the maybe more relevant topic for age disorders. And unfortunately, I think we will leave with probably more questions than we started with, but I think it's a good starting point. And I think I see many familiar faces, pioneers of the RNA polymerase III field. So I think it's really a great opportunity to start like this. So as we all know, what you see here depicted in this actually original picture of the transcript of a lecture from Francis Crick is what is considered the central dogma of molecular biology, which really states that the genetic information contained into the DNA is translated into a final product, into a protein, passing through an RNA intermediate. This process is called gene transcription, and as we now know very well, is performed. This task is performed by 3 very well specific eukaryotic enzymes: RNA polymerase I, II, and III. And again, I really like always to show this slide, which is these were the end of the 60s, where technical equipments were certainly not as advanced as we have today. And every time I see this graph, this is a purification of the 3 eukaryotic enzymes, I'm still in awe of these pioneers that made it happen already 50 years ago. We struggle these days to obtain this type of purity, and so it really means that we are really working on a field that has been seen really pioneering figures to be set by really pioneering figures. And so we are interested in general, specifically on POL III in my lab, and this is also because POL III is involved in very important physiological function that affects health and disease. POL III, I see that Nazif Alic is here, and he is one of the first to really discover that POL III is one of the most important determinant of aging, and this is a function which is conserved throughout the eukaryotic system. There has been always let's say the finding of augmented POL III transcription in many forms of cancer, if not all forms of cancers, and there has been always a question of a possible causative role, and we have partially contributed to this for a specific subset of POL III gene to understand what's going on. Of course, I mean, in this specific setting, I think I don't even have to remind how important are inheritable mutations of POL III that affect a variety of neurological disorders or diseases, including 4H syndrome and many others. And this is something that, as I said before, it impacts a very, it's pediatric patients, right? So I think is like one of those fields that we are more sensitive to, because life of these young patients should not be, in a way affected the way it is. So I think it's good if you could do anything to really ameliorate their conditions of living, or even, having the dream to really cure these diseases, which are very difficult, but like we will see. Another aspect which is, I think, very interesting also on POL III is the relatively recent discovery of an involvement of POL III in native immunity, or more specifically in a function of DNA or viral sensing in the cytoplasm, where it seems that is the unique polymerase that really can transcribe in the cytoplasm foreign DNA, eliciting a very, very strong immune response. So in my lab, we are interested in understanding very general basic mechanism of POL III transcription. In the last years, there has been an explosion of mechanistic studies, and this is great. We understand it better. We are still trying to understand really how this process is regulated by several effectors, and in general, it's really the idea is try to dissect how the transcription of POL III genes really affects cancer in particular. I think it's clear that tRNA biology is way more complicated than originally thought. One, is not just about having these very long living molecules produced, but there is a lot of regulations going on. tRNAs are also substrate to be, trimmed and forming very, very, very important signaling molecules. So it is something we are looking actively into. And we are also interested in what is being defined extra transcriptional roles of POL III, within general, let's say, deal with the supposed role of POL III and accessory factors into genome, eukaryotic genome functional and spatial organization. I won't talk anything about this today because it really sort of drifts away from what we know about the 4H syndrome, but there are some interesting aspects of this biology as well. So going directly diving into the topic, really what you see here is a comparison of the three eukaryotic polymerases. The nomenclature here is the east one. Unfortunately, nomenclature is an issue. It would be much nicer to have something a little bit more easy to follow. I just reported here the nomenclature for the various subunits of the human version of polymerase III, which, as you can appreciate, is the more complex of the three eukaryotic polymerases. Is composed of 17 subunits, accounting for around 700 kilodalton. And again, this is like something to remind ourselves, or just to make the point clear, is that the three eukaryotic polymerases are somewhat related. So there are subunits which are indeed shared. Some of them have paralog functions. And one major difference is between POL III and POL II, which is, let's say, the mostly studied and the better understood in a way, is that a set of what are called general transcription factor in the POL II systems became accessory, permanently bound, stably bound subunits in the POL III system. So if we make a comparison between POL III and POL II, it's like seeing POL II with a set of stably associated transcription factors that of course confer certain specific function to that polymerase. What I recapitulate here is the architecture of the type of promoters, of the type of genes that polymerase III transcribes, because for accurate transcription, the polymerase does not act by itself, but requires the activity of a series of general transcription factors. And again, this is another case where these were sort of schematic representations based on the analysis of very few selected genes back in the days. And it's truly amazing how these results still holds true now that we have, you know, the complete genome sequence, and look how these factors have like occupy different sites in the genome. It still really holds true, again highlighting how strong were the basis of those pioneers that studied these things like 50 years ago. So we are still, you know, doing our studies based on these initial discoveries. And so what is really important here to highlight is that substantially the activity of POL III ultimately relies on the activity of a transcription factor called TF3B. The other factors like TF3C and SNAP-C are very, very important, but they can be really regarded as recruiting factor, is what bringing the polymerase there. But these represent really the minimum set of proteins, of protein complexes, this is required to start faithful transcription. I want to just highlight this class of promoters here because we have been working quite a lot with that. It's something which only exists in vertebrates, so you know, the human system, and I think is quite relevant because we will see how the human system actually is way more regulated and evolved when compared to the yeast system, despite the general conservation of the architecture of these enzymes. So the message that I will give you towards, or in the middle of this talk, is that it's true that there is a lot of conservation between the systems, so we can derive a lot of useful information, but based on the yeast system. But really looking at those things in the human system is really relevant to understand, especially to understand diseases, but also to understand layer of regulations that do not exist in the yeast system. Speaking of which, yeast, as I mentioned, has been our elective organism for studying this, for studying RNA polymerases from a structural perspective. And this is because it's relatively easy to obtain a lot of biomass. So those proteins, those complexes are not very abundant. We need to get a lot of them to subject them to structural analysis. And so yeast has been one of our favorite organisms, still is, where we can purify very nice complexes like the one depicted here. Another thing that I want to just mention is, I mean, I'm sure all of you are well aware of what is called the resolution revolution that happened in the last years in a technique called cryo-electron microscopy, which really has become the elective technique to obtain structural information on protein complexes. So, and at this point, I would say that it's clear that in the next couple of years, you know, X-ray crystallography, which was the elective technique before this revolution, will be really confined to a minority of projects. This is because EM has kind of caught up in terms of the resolution that can be achieved. You could see how before 2013, I'm not going into the details why, but the details that you could get about a certain complex were very fuzzy, very blurry. It was called blobology back then. I did my postdoc already on, on RNA polymerase III, and yes, we would see more or less well-defined blobs. And these days, we basically can really obtain atomic models, right? So this is a great advancement because we need much less material. We can treat a broad range of molecular weights or size of complexes, but even the more important thing is that we can tackle complexes which are conformational heterogeneous. This was obviously a bottleneck for crystallography. We needed highly purified complexes, but also purified in the sense of the conformation in which they would be. In EM, this is, in most of the cases, can be dealt with by computational approach. And so what we do is basically recording shadows of this protein complex at the individual level, individual particles, once they are frozen in a thin layer of vitreous ice, as you can see here. And then what we do is extract, box all these particles that have a very, very low signal and do what is called averaging. So basically putting them together in stack of views where we have proteins or molecules with the same orientation. And by this process, we really enhance what is called signal to noise ratio. So we can have really, really high details of these individual views, and then combining them, knowing the angular relationship between these views, we can obtain very detailed three-dimensional reconstructions of the complexes under study. And so we have used this approach initially using the yeast system. Christoph Müller's lab was actually the first to come out with a very nice atomic model of the yeast RNA polymerase III in its apo form and elongating form. And we have applied this to study what is called the minimum pre-initiation complex. That, as I said, give us an idea of how POL III transcription initiation occurs and is regulated. What you see here is a EM map where we built the model of this kind of basic pre-initiation complex of POL III, and I'm not going too much into details, but we really code the polymerase in the act of transcribing a gene. So the DNA was loaded into the active site, open, and this was all done by the polymerase, by the enzyme itself. So we didn't use any sort of trick to stabilize it in that way. So, again, this is one of the advantages of EM, and so we could come out with a nice model and explain what was known already in the literature from biochemical experiments, where we could really show how accessory transcription factors would play a double role into recruiting the polymerase at specific genes, but also in a way activated or prime it, for initiations by stimulating the DNA strand separation activity, which is required really to initiate transcription. And thanks also to the efforts from many other labs, Christoph Müller's lab particularly has worked both on POL III and POL I, Patrick Kramer's lab on POL II. There are like several labs now working on these mechanistic studies. We could really have a look and compare what are the mechanisms used by the three different polymerases. And one of the conclusions was really that POL I is really the polymerase that more deviated or drifted away from a common ancestor, if you want. This kind of makes sense because POL I really transcribes one specific gene, so it can sort of drift away because it will co-evolve with only one gene. While POL II and POL III are more related, as you see from the similarity of the position of these factors, which are colored in the same way, and of course with their own with their own specific mechanisms of regulation. And one of the things that is important for POL III is that it's a very efficient transcription machinery. And one of the reasons why it's so efficient is because throughout interactions with this TF3B complex, these complexes are incredibly stable once bound to the DNA. They are not just stable and specific, so in a way that POL III is recruited where it is required to transcribe genes, but it's also, as I said before, primed for transcription. So everything, once POL III is recruited by TF3B, the DNA it's capable of opening the DNA spontaneously, and so it just needs nucleotides to come in and start productive transcription. So there are no any more structural arrangements required to effectively go into initiation and elongation. And what I mentioned before is just, I want to point it out here because, as I said before, the human system compared to the yeast that's been a valuable system is much more complicated. One example is the existence of this alternative TF3B complex in the human system, which we're very interested when I opened my lab to understand, because in this complex we have a component called BRF2, which is not existent in yeast, is related to the common protein called BRF1, and really in human transcribe just a very, very small subset of genes. And we really ask, why is that the case? Also because this protein BRF2 is very often amplified and overexpressed in many forms of cancer, and there is a strong correlation between copy number variation and expression, suggesting that this type of cancers, in particular breast cancer or lung cancer, seems to be really addicted to high level of expression of this protein. And so what we used there was classic X-ray crystallography. In this case, we solved a beautiful structure, and I'm gonna skip it towards the end. But what we discovered there was a functional element, which we discovered to be an element that gives this subset of gene the possibility to be regulated in a redox-dependent manner. So they can be switched on and off, let's say, in case of oxidative stress. And this is the case. So we did a couple of experiments to show that the bulk of POL III transcription, it's really not dependent on oxidative stress. So this is the level of transcription of a classic tRNA with and without oxidative stress, while products that are under control of BRF2 are really oxidative stress dependent. And to make a long story short, we just had this model, which we still have to fully prove, but it gives an indication that there is one of the reasons why in cancer cells we have overexpression or amplification of BRF2 is in order to sustain the synthesis of selenoproteins, which are proteins which are very important for oxidative stress detoxification of redox homeostasis, because cancer cells really thrive in oxidative stress conditions, while normal cells would not do that. And we believe that BRF2 could be actually also work a little bit as a master switch for normal cells to really switch off and go into apoptosis when oxidative stress is too high. And I'm just mentioning this here because I think it's always good to remember, if you are looking into therapeutic approaches, that there are also part of POL III transcription that might be affecting other very, very central or fundamental cellular biological processes. And so we are still looking into that and looking into more complex structural biology approach as well as functional approach to understand better whether BRF2 could be a novel target in cancer. But having said that, this was just an example, a little bit to showcase what we have done in the past, but also again to highlight the fact that human is more complex than yeast. That was a case that exists only in human. And so it's clear that we now have to look at these complexes in human. And so our approach which was very similar to what Christoph has done, even doing a different cell lines, was to use CRISPR-Cas9 to genome edit cell line, in this case HeLa, to attach basically an affinity tag. In this case, it's a GFP, so it serves a double function, to use it as an affinity tag, but also a fluorescent tag to visualize these complexes in living cells. And this, you know, again, it's not easy. It can be done. These days, they all, you know, you hear people say, just use CRISPR-Cas9. It's not that easy to do it in a lab setting, even for research purposes. It doesn't work all the time, and it's quite painful to do it. Anyway, in particular, in collaborations with Christoph Engelslag in Regensburg, they managed to obtain this really nice cell line where we tag a subunit which is shared by POL I and POL III. And what in fact you can see here in these cells, this magenta is DAPI staining, which highlight the nucleus of these cells. You see that there is, in general, kind of an homogeneous green color in the cytoplasm, here and in the nucleus, and then some enrichment of this more dense part, which are the nucleoli. So because this is a common subunit, we believe that this is mainly POL I, which is usually found only in the nucleus, in particular around the nucleolus, while this homogeneous green staining in the cytoplasm and in the nucleus is probably only POL III. And in fact, when we do fractionation experiments, so we purify the nuclear fraction, you can, using as a tag GFP, using a nanobody against GFP, we pull down both POL I and POL III, while when we purify this just from the cytoplasmic fraction, we get only POL III, which again, sort of support the idea of POL III being actively accumulated also in the cytoplasm and not just going directly into the nucleus as for POL I. We can obtain a really nice sample, which is pure and also active. As you can see here, this is an RNA extension assay where you basically feed this substrate to the polymerase and let it elongate. Those are elongation products. So we were quite confident that we could, we were seeing, we should have purified the functional complex. And we, again, you employed cryo-EM to obtain a structure of what is called the APO polymerase. So the polymerase without being functionally linked to either DNA or to transcription factors. And you know, it was remarkably similar to yeast in general in terms of general architecture. And I must remark, as we have already mentioned, there are actually not just 2 papers, but other 3 papers describing this. And I have to be honest, you know, our reconstruction is probably the more limited when compared, certainly when compared to the one with Christoph. They obtained much better resolution, much more homogeneous map that really covered areas, also functionally important areas of the polymerase. And this is because they had elongating complexes which are more stable. The overall structures are the same, but I would suggest that if anything, anyone is interested in looking at very detailed interactions, etc., to use Christoph's models and not ours. Not because they are not bad, but their models are definitely better and more complete. What we were interested, and maybe this is something we have done slightly different than others, was also to look at a specific part of the human polymerase, which is really not conserved in yeast. One of the major differences is the presence in human of this long C-terminal extension of subunit RPC5, and we were really puzzled because it seems to be quite conserved. And so we studied the structure of this by X-ray crystallography. This part, in reality, has been resolved by EM, again by Christoph's group, and there is a very good agreement with what they see in EM and what we see in crystallography, but we also solved the structure of this other tandem with helix domain. This doesn't tell much. Besides, we also employed another technique to have an understanding of how this whole extension will look like. It would probably look like something like that. So these 2 or a couple of tandem winged helix domains with a very flexible linker in between. And you know, we were trying to understand what are they important for. And in our quest to understand that, this is not, you know, is far from being final, but we basically knocked down the endogenous RPC5 and sort of supplemented or ectopically overexpressed versions of RPC5 lacking this C-terminal extension. And we didn't see much difference. It's not a very clean experiment, but we didn't see very major differences in terms of global transcription or in BRF2 dependent transcription. We were thinking along those lines. But what we saw basically was the fact that, and I'm not going into the details, it seems that the C-terminal part of RPC5 is really important for the stability of the enzyme. So when we remove this part, the enzyme seems to be much less stable than the wild type one. And it's not just the RPC5 subunit per se, but also other subunits of POL III that are affected by this. And so this again stress the fact how this can highlight another layer of regulation presence in human, which is not present in yeast. We don't exactly understand whether this is a process that actively requires, for example, proteasomal degradation, but we are looking into that. The most relevant, of course, for this seminar was also to use this structural analysis to really map the variety of mutations that have been reported by all these genetic studies during the years. And I must say that we had already pretty much a good idea of what they were doing by using the yeast system. But now I think having these really accurate models of the human system, it really in a way validate even more what we already understood. And the main message there is that most of these mutations for all the, I would say, to generalize, all the neurological disorders are really mutations that involve a level of loss of function of the enzyme. And this is said in a very broad spectrum. There could be mutations that are very, very detrimental and even impair biogenesis of the enzymes, and mutations that are more let's say tolerated, and so that they will probably just an effect on global level of POL III transcription. So what is interesting, I think, and again, I think Christoph's lab did a very good job on putting all these informations into their models and maps. And I think, again, if you really want to look at this into details, I strongly suggest to use their models. I think what is clear, and this is coming from our analysis, Christoph's analysis, and other groups, is that these mutations, what I find fascinating is that they're not like spread all over the place, but mostly tend to form this kind of hotspots, right? And this really indicates that there are probably hotspots which are really important for the general function or stability or biogenesis of these enzymes. And in fact, the mutations that we have mapped mostly either abolish or disrupt the function of core elements, or they are, for example, placed at interfaces between subunits, so that the enzymes is either not formed correctly or cannot perform its function correctly, or is much less stable. Now, this is very difficult. I mean, in general, there is, I think Christoph's put it quite clearly in their paper, there is a general correlation between, let's say, the clash that those mutations causes and the phenotypic effect or the severity of this disease. I think it's difficult to really say that for each single identified mutation that is the case, but in general, I think it's a pretty good approximation. So the more disruptive this mutation is in terms of clashes, the more phenotypic effect we see, or the more gravity is observed in these patients. And of course, the Tricker-Collins syndrome, which really affect biogenesis of these enzymes, is in fact, in general, probably are mutations that are more severe. And in fact, they are the great majority of them, if not all, I think they are dominant, while the majority of mutations of the 4H syndrome, if I remember correctly, are actually recessive mutations. So the general really idea here is those are mutations which are that are affecting the core of the enzyme. They have a very broad spectrum, modulating, so sort of like almost doing a gene dosage effect. This is not. There are also, I'm pretty sure, certain mutations that affect the general expression of some POL III subunits, but and this makes it, of course, very difficult to approach them all in the same way, because at a certain point, these different disorders might turn out, one might turn into each other. It's a very difficult spectrum that we are looking at. What was interesting was also to look at those mutations that give rise to viral susceptibility, so hypersensitivity in this case of varicella virus. And in general, again, those are very much peripheral, so they don't affect the general function of the enzyme, which kind of makes sense because these individuals are perfectly healthy unless they are hit by, and I don't even know if all of them, but unless they are hit by varicella virus, in which case they really develop very severe symptoms when compared to a normal individual. And this is probably due to the fact that these mutations are affecting parts which are very likely affect the general DNA binding activity of POL III. So, which again, in the cytoplasm is probably driving transcription of this foreign DNA without using accessory factors, so relies on transcription just by its inherent affinity for double-stranded DNA. And so this kind of makes sense. Having said that, I think I'm going really towards the end of my talk. I didn't go into really describing any single particular mutations. I think it's plenty of examples. I think what I really wanted to do is try at least, and I'm not an expert again, I'm sure that there are more qualified people in the audience that will help you guys defining what are the most pressing questions and where really to put the resources we have. From my perspective, I think there are still a lot that we don't understand, despite we can now map these mutations. So we don't know, for example, if the symptoms and the disease that we see in 4H are due to a global tRNA deficiency, or is maybe due to the fact that maybe one or few specific tRNAs are missing in a specific time frame during development. We also have to understand, I mean, for me, it's difficult being somebody that works, all in vitro to understand that, during development, certain mechanisms might be really fundamental and finely regulated at a specific time, and then might become much less relevant once once an individual has completely or fully developed. There might be other non-coding RNAs involved, still POL III transcribed. Another thing that I'm putting there, POL III has been recently discovered to play an important role in DNA damage repair as one of the effectors that really transcribes RNA at the site of double-strand break, and I think that it is also probably something that one should consider. There are many neurological disorders that are indeed that see mutations in DNA damage repair proteins, so it's a link that maybe one should also explore. From the tools that we are missing, I don't know. Again, from my perspective, a system where we could relatively easily or in a high throughput manner try to screen the phenotypic, let's say the phenotypic effect of these mutations in a reproducible manner would be very much needed. I'm thinking about patients that have organoids. Again, this is not straightforward, but it is possible to do it in a high throughput manner. So you use cells from patients and sort of develop mini brains, if you want, in vitro. Of course, they do not recapitulate the entire process, but they are good surrogates before going into animal models, which, you know, we are still, you know, missing. I mean, I know that Robin and Ian did a very, very good job at having a conditional mouse model, which is already very valuable. But again, this requires a lot of time, a lot of efforts, and it's not easily scalable, while maybe, you know, organoids could be probably something more treatable. It's this kind of in between in vitro, in vivo system that could be very, very helpful. Also because as biochemists, structural biologists, we can go back and use those really as platforms for you know, extracting information. So, you know, looking at POL III, looking at single cells, etc. So this would be very interesting in my opinion. And in regard to therapeutic intervention, I think I've asked a little bit Valerie already in which directions you guys were thinking, but, and of course, you know, again, genome editing, maybe screening stabilizing compounds would be a great idea. Another thing that came to my mind, and you know, this is used sometimes in cases where you're dealing with you know, a process which is an housekeeping process, is to use the concept of synthetic viability, right? Originally, what was discovered in yeast was this concept of synthetic lethality. So, you know, modifying two things that are not lethal per se makes the two things together lethal. This is the reverse, and say, okay, if something goes wrong in the cells, can we modify something else, maybe apparently unrelated, to make the original thing right? And I don't know if this will apply to this specific case. But I think, again, if we would have a system like, you know, patient-derived organoids, it wouldn't be that difficult then to do you know, genome-wide screening of other factors that can be turned down. It would be exciting if you could, you know, discover that maybe by inhibiting a specific tRNA synthetase or something related, we could at least ameliorate some, maybe slow down the entire process, but ameliorate the patient's outlook, for example. This, of course, is, you know, sort of pipe dreams, etc. And with that, really, I just want to thank the people that you know, had the faith to join my lab. Now is what, 9 years ago. It feels at the same time really short, but also like much more than 9 years. And especially I want to also thank my collaborators, Christoph, with whom we have developed this CRISPR-Cas genome edited cell lines. I want to take the occasion to really thank my, you know, competitors. I mean, Christoph is here, and I have to be honest, you know, if I would have to pick a competitor, I was really lucky to have him as my main one, because we really, besides people, we get along well, but he's, you know, we try to tend to make both aware of what's going on, and is, I think, the way science should work, also for the benefit of the people working so hard for those things. If I can spend just other 2 minutes, I don't want to self-promote the institute, etc. I mean, besides it's a beautiful building, but I just want to mention this because initially we would have 5 major centers. Centers, you know, we are all in the same place, so you can call them centers or departments. But I just want to point it out that so the second best after structural biology, which I'm directing, is going to be the neurogenomics center, right? And so this is, I want to mention it because the director of the center, Giuseppe Testa, is really one of the world leader in using pluripotent stem cells from patients to derive organoids. And so we are planning to have a facility, if you want, to automate this process to create brain organoids. And of course, I'm in discussion already with him and see whether we could bring him and colleagues maybe on board if this is deemed, if it's something that is deemed to be important to look into foray or POL III related leukodystrophies and use their expertise to really tackle this problem. And with that, I just conclude. I thank you all for your attention, and I hope I'm happy to answer your question, but also, I hope this will ignite some interesting discussions on what to do next. Thank you.
Well, yeah, I was just about to say, I will now turn it over to Sébastien, who will lead the discussion because he's the one who knows what he's doing. So yeah, and he already said, please put your questions in the chat.
Yeah, right. Thank you. Thank you, Alessandro. Maybe we can go into 2 rounds of questions. Maybe some people have general questions about your talk, and maybe we can focus later on more dedicated questions about 4H and what you already mentioned at the end of your talk. So don't hesitate to raise your hand. So I don't know if it works well on Zoom, or ask the question on the chat, and we can re-ask or give you a hand to ask directly your question to Alessandro, or maybe Christophe, because Christophe is also in the audience. So please, let's go. Does anybody have already a question for Alessandro?
I see a couple of hands, Sebastian. I think Professor Willis raised his hand, and maybe then Bob White next, please.
Ale, thank you for the great talk. Great to see you and everyone else. So my question is a structure function one that sort of relates to your finding and others, that the mutations that cause POL III related leukodystrophy tend to be clustered in particular regions of the polymerase based on current knowledge. But you know, there are still many new mutations being identified in patients, and many other subunits of the enzyme that have not yet been targeted by mutations. And given that the nature of the disease, for the most part, but not exclusively, involves loss of function, it seems that the potential for targeting many other sites in the enzyme that are going to compromise function is quite large. And so the structure function map is going to expand significantly over time, I would predict, and I'm interested in knowing what you think about this.
Yes, it's a very good point, and certainly you have a point. Again, not being a geneticist myself, and you know, yes, I presume that that's the case. Probably there are, you know, those hotspots are the first one to come because they are probably the more obvious, or if you want, places where mutations tend to accumulate, and probably they have a prevalence within the population that is higher than others. But you know, you probably have, like you say, the spectrum of very low abundant, you know, mutations that are not flagged because they are at so maybe low level of incidence that is more difficult to characterize. I also don't know whether, you know, there are other factors, well, genetic factors that contributes towards, you know, the outcome of those mutations. So we identify this mutation as causative, but you know, we don't know whether that only mutation is responsible for the entire phenotype or the entire pathogenic, let's say, potential that you see. So that's also another problem. But yeah, you're right. So we probably have a full spectrum of other mutations.
Okay, thank you.
Apparently, Bob White would like to intervene. So if you...
Hi, Alessandro. Good to see you.
Good to see you, Bob.
Thank you for your talk. So did I understand correctly that the model with regard to the varicella zoster is that they have that the polymerase mutants have a weakened affinity for DNA and therefore mount a less strong response to the virus? Is that the idea?
That's the idea. Whether it's actually the case, I'm not 100% sure, but it would point towards that direction, yeah.
And is there, are the mutants able to bind nuclear DNA at normal rates? Has that been looked at normal affinities?
Sorry, say it again.
Well, I was just wondering if the affinity for DNA is compromised, is that specific to the virus, or does it also have a compromised affinity for nuclear DNA?
I think there is nothing special about nuclear DNA versus viral DNA in that respect. What I think it changes is that in the nucleus, the activity of POL III depends on all the auxiliary transcription factors, and it doesn't. So to put it blunt, my idea is that really in the cytoplasm, POL III find a blunt end double-strand DNA, it can bind to it and transcribe it. Well, you know, this obviously doesn't happen in the nucleus, and for that activity, those mutants probably are lowering a little bit that type of affinity in a way that is not an efficient process anymore. In fact, what we see from yeast structure, elongating complexes, and you know, Christoph can confirm, compared to other polymerases, POL III has a very, very strong affinity for double-stranded DNA, even before it opens or whatever. So in the clamp, it holds really tightly to those blunt end double-strand DNA, and we know it can even open it. So just even without having, let's say, you know, transcription factors involved, we identified one of those intermediates in one of our reconstructions from yeast. So which I suppose is going to be the case in the human system as well. Other polymer, and it has maybe a lower affinity for RNA DNA hybrids, so during transcription, that's why it terminates so efficiently when compared to, you know, for example, other polymerases. So it's a difference between those polymerases. But
so in the nucleus, the presence of TF3B overcomes a weakness in interaction with DNA.
Yeah, I think it's also the point where, you know, in the cytoplasm, I think the DNA binding occurs through recognizing a double-strand blunt end, which in the nucleus you don't have. You have, you know, you start from being recruiting from TF3B, and then you have to load. So the mechanism of loading would be probably different. While in the cytoplasm, if you have a blunt end double-strand DNA, you can imagine that you can also just load it directly by going in the clamp. And in fact, in one of the reconstruction of these other groups, so they even found one of the apopol III bound to some, you know, endogenous double-strand DNA. Just to tell you how much. So they brought with them some double-strand DNA from the purification. So I think it's a thing.
So on that basis, you might expect those mutants to also be compromised in homologous recombination based on the recent discovery.
Very likely.
Thank you.
Very likely.
Thank you.
So there's a question from Vivi Einer, please, Vivi.
Hi, thank you. Thank you for your presentation. As maybe more of a kind of a remark, I'm the head of the stem cell lab in Amsterdam at the VU University Medical Center, and I closely work with Dr. Nicole Wolf, who is seeing a lot of 4H patients, and we already have a research line ongoing using iPSCs from 4H patients. And we have another focus. We work more on studying how neurons and oligodendrocytes are involved in cellular mechanisms, but we are generating different kinds of iPSC-derived cultures, and we also generating organoids. I do not know to what extent you need them in a high content. That worries me a little bit, but I just want to indicate that as these cell lines are already around, and this is not our research focus, I would be happy, and of course I would involve Nicole, whether we would be able to share the patient lines or not. But I just want to indicate that we have maybe already a first start to bring this kind of research faster.
Yeah. Thanks for the remark. And mine, I didn't even really specifically talk internally in order to develop something like this, because I think, like I was talking to Ron, Valerie before, I think, like, we don't have, so there are like so very few resources to study those things that we really, at the level of basic research, academic research, we cannot really afford to compete with each other or even to replicate things that have been done. So really, structural biology is different, but in terms of resources to create cell lines, etc., we really, it's great to have this table here and to discuss these things in order really to divide and conquer, if you want, right? Divide efforts, because it would be a shame to not to do it in this way. Ultimately, we really have to do, as basic research scientists, our best really to drive this for potential benefits. So
no, absolutely. So that's why I would like to indicate that we already have maybe something ongoing if people are interested to pick up these studies.
Sure.
And I'll just jump in. Thank you for that, Vivi. That's the kind of collaboration and sharing and getting resources together that we're dying to see. And so, we're all making it. Christina and Valerie and I are making note of that comment too. And we'll also circle back after this talk to maybe see if there's an opportunity for a collaboration there. But thank you so much for raising that.
You're very welcome.
Then Christoph. Hello, Christoph.
Yeah, first, thanks to Ale for this really nice presentation of the POL III and actually also acknowledging so nicely our nice collaboration or friendly competition, let's put it this way, hopefully to the benefit of everybody. I still want to come back to these questions of mutation. So obviously, as Ian said, there seem to be a lot of mutations which affect POL III structure, and that's probably also because it's a very intricate complex. There's a lot of protein interfaces, quite sensitive to disturbing the architecture through mutations in the core, and then everything just falls apart. But this is more generally. I was wondering, how good is the databases? How much SNP's mutational data are really known, which one can map back to the structure, and especially also mutations which don't have phenotypes. People who one would declare as completely healthy and still have mutations in the polymerases. Do you have any insights about this? When there are mutations, some seem to be have more stronger effects, or perhaps also depending on different patients, since other patients have much lower effects. But are there also mutations which hardly have any effects? And so how good is the databases there?
That's obviously not a question for me, but it's a more general question, I hope.
It's a more general question, yeah.
I have a little insight about this. In general, looking at human genome data for POL III subunits, particularly the largest ones, there's an extraordinary number of polymorphisms in this gene. And so I think there is a large number of missense mutations that are out there in the population that are not appreciated in terms of their potential impact on function. And so, it's only in these unfortunate situations when a child happens to inherit two alleles that have these negative consequences that we realize that there's a disease outcome. But I think we've only scratched the surface of understanding the impact of mutations in the human population on the function of this enzyme.
So it seems that there's no more dedicated.
I have a question.
Oh, yeah, sorry. Please go ahead.
So just following on that, does anyone have a feel for how much the sort of wild type POL III activity is in relative excess? How much do you have to lose before you see any sort of phenotype? Is that even known in yeast to begin with?
Well, there are some insights about this, and in the mouse system, at least, I think we can point to some recent work from Bob Rader's lab, for example, where they've knocked out the G and GL paralogs and find that to get through embryonic development, you need more than 15% of polymerase activity to pass through gastrulation, because these embryos die somewhere between E3 and E6.5. And based on the amount of the subunits that are present, they've estimated, or they have data in their paper that allows you to estimate approximately 15% activity is needed to get through embryogenesis. After embryogenesis, I think it's less clear how much you need, but clearly you don't need as much. Embryogenesis seems to be more demanding on the amount of polymerase.
So did I understand you correctly that 85% is surplus even at gastrulation?
Well, I guess you could interpret it that way, although I would say, you know, requiring more than 15%, I mean, that represents the low bar. You need to have more than that.
Right.
Exactly how much more for normal function is not clear.
And in yeast?
Not really sure there. I mean, I can recall an early paper from David Brau's lab that indicated something like 20% of U6. If you have less than 20% of U6 snRNA, a yeast cell is dead.
And probably then it also changes by which type of conditions, you know, you are looking into.
Sure.
I assume, but.
Thank you.
So if you focus on the 4H multiple mutations, I was wondering, I mean, through the discussion and the huge efforts that would have to be made if one would like to understand each mutation work, if there would be from let's say the molecule basis point of view, a short list of mutation to look at, and on the other side, on the, let's say, pediatrician or neuroscientist side, a particular point mutation to be identified in order to just to focus or to come with a really a short list of things that could be done. Is that is there a sense in that?
Sorry, you mean to sort of rank?
Yes.
Well, I presume one. I mean, those are very, very rare mutations, but still probably within those you can still find the one that occurs more frequently if you want, or at least group them in a very similar position. I guess that would be. I think, you know, I think what would be really important is whether we could imagine to have sort of an approach that would hit some of them, at least that are kind of like, you know, structurally, functionally related. And so I don't know whether it makes sense to choose some exemplary one that might be even different in terms of what we could predict structure function would be. And so that would cover more. So I'm pretty sure, you know, I don't have really data, but I think biogenesis of these enzymes has been completely overlooked. We don't really know how it works. I mean, there are a couple of tough factors that we know are pulled down. And this is, you know, this is probably also very important. At certain stage, you would have probably mutations that impact how certain subunits interact with the chaperone or you know, and so I think it would be good to look into those as well. But again, from the perspective of being proactive, if, you know, let's say pediatricians or, you know, medical doctors would come up with just like, okay, we really need to understand this mutation, that mutation, that mutation. It's certainly an effort that we could do, or already on data that we have. And if something else is required, like, I don't know, can you check whether, you know, DNA binding affinity or versus, I don't know, transcriptional output in vitro is something potentially can be done. So that's maybe also, you know, something good to take on these 4 studies that came out. Christoph and myself use an approach which use CRISPR-Cas9, and this is tedious. It takes a long time to generate the cell line. So you can't do really mutations that easily. On the other hand, the other groups use more like, and you know, it's recombinant basically polymerases by, you know, overexpressing, you know, from plasmids. So in that case, you can think about creating ad hoc versions of enzymes that one can purify that harbors those mutations if it's needed to be. I think for the most cases we can really predict what the outcome would be. But again, if we want to really at certain stage have, you know, data and say, look, these enzymes is less stable. Sorry, this enzyme is less processive, or it's, you know, for whatever reason slower. I don't know. In principle, we could do it. Obviously, we can't do it on 500 on 500 mutations. But if we really identify a couple of them, you know, it is possible to do it.
Yeah, perhaps just to say that I would support its view that one would rather probably look for types. I mean, are there some folding types or activity types, and try to characterize those, and if one can classify those, that would already be quite helpful in a way that one really says, yes, this is now more the stability of the whole machine, or this is really the activity, or this is just interaction with another factor, which seems to be like with nucleic acid with the viral susceptibility. And I think that would be a good approach.
That was the point, to kind of cluster mutation to facilitate the analysis. If we needed, I know that the point mutations are really rare events, and unfortunately, but that's from, let's say, structural biology point of view, that's what one can propose. And I know that on the other side, on the medical aspects, it might be a bit different, and the need or might be a bit yes, differently seen. That's why the aim of this association is to put together all the people so that discuss and share their experience and their feelings about how to handle, how to go. So I don't know if Valerie would like to say a few words about or maybe Ron, about the roadmap that they managed to propose at the end of the last November meeting. Maybe this could be of interest for Christoph and all the Oddpoles community, I would say.
Yeah, I can say a little bit, but so our endeavor, we basically just had, as Ron mentioned, our first meeting to think about what the roadmap would be, and where the efforts should be. And one major sort of tranche that we are thinking about, which is why we have these kinds of meetings, is to really, you know, find what the most pressing questions are. What are the questions in basic research that we should pursue? And we hope that this will sort of come out of these meetings. What is it? I've heard a lot of interesting things today that sort of made me think about in my, direction, like, is this what is the thing with the issue of DNA repair? I hadn't thought about it. What do the non-pathogenic mutations, can we learn something from their function? What can we do? And I think one of the things we want to do is hear from all of you what you think is important or questions that should be addressed. And then think about, whether we can provide some kickstart funding for these projects just to get them going. Another big goal, as we mentioned before, is sort of bring people together. And we've seen already the good examples, Vivi says, yes, we have the cell lines, if you need them. Because as Alessandro, you said that too, the resources that we have in terms of funds and time and things that we can do are limited. And as we come together and talk about these things, we might be able together to kind of accelerate things by knowing that. This is sort of the things that we want to do in basic research. Of course, we are interested on promoting a mouse model because we know how important that is. There's actually, work going on. We have talked about organoids. Once again, we haven't made up our mind yet. We are, of course, there are other tranches that are important, like patient registries and natural history studies that we're working on trying to initiate or, support the current efforts. And there is an effort to look into gene therapy. So that's just an overview what we think that we will do. And with that, I don't know whether people have more questions. Otherwise, I think I would like to thank everybody for joining us today. I would like to thank people for great discussions, Alessandro for presenting. And Ron, if you would like to have some last words, I'll hand it over to you.
Sure, yeah, and thank you, Valerie, for your great work. Charting our course and direction in science and engaging with this great research community and on this seminar. So thank you. I just wanted to really add two things. One is Sebastian and Valerie just mentioned the research roadmap. The roadmap is available. It's been published on our website, and we'll share it with all of you after this call. And what I really want to underscore is that it's a version 1.0, and it's a dynamic, living, breathing document that we want to update and continue to evolve. And so we invite you to review it, to share your thoughts as to your own ideas about what changes and additions to make. And so we'll circulate that after this call. The second point I want to make is really just a personal one as a dad, and to tell you that this kind of convening and collaboration and focus on our disease area, and frankly, as I hear your comments, much of which I haven't even been aware, we haven't been aware, is extremely, it just provides me with great hope. And I know that I represent the entire 4-H sort of family and patient and parent community in saying that this is really exciting to all of us. And so, I want to really thank you sincerely and deeply for all of your great work. And maybe at the risk of, sort of pre-announcing something that we're not yet ready to announce, what we're working on, and I think Alessandro mentioned, the desire to get together in person. And, convenings like this are so valuable. We, of course, all hope that getting together in person is something that we'll be able to do safely again soon. And we're working hard on planning, what we plan and hope and intend to be an in-person convening sometime in early 2022. And so we'll be reaching back out to you as we work, we're working on details around that, but I do want to just put that out there now that, we'd love for you all to join us for that. And frankly, we'd love for you all to be involved in helping us prepare for that and charting the agenda and looking for opportunities to present all of your work and continue this discussion at the convening. And so with that, I'll just thank you again for all the work that you're doing and the hope that you're providing to us. And we very much look forward to continuing and growing our collaboration together. Thank you all.
Thank you. Well, thank you, Ron, for involving us all also in this. I think, you know, it's a little bit of a, not a dream, but, you know, it's good to feel as a basic research scientist to try to give back and not just doing this as an exercise of learning something, but rather, you know, try to give back. So thank you.